So once again, thank you very much. And I'd like to hand over to uh, Mark Tishkovitz, who's going to tell you about the PTEN registry. Thank you.
Great. Good morning, everybody. Thanks for the introduction, and thanks for having me back here again. So my name is Mark Tishkowitz. I'm based in Cambridge. There's a small team of us. I will introduce them shortly. How do we go forward? Is it just this one here? There we go. Yep, fine. And I'm going to talk to you about the patient registry that we've been running quite a long time now. That has been funded through the PTEN Research Foundation, and these are the key team members. I think they're all sitting at the back. I don't know if you want to raise your hands as you go along. So, Catherine, you want to say hello? Yes, and Angie. There we go. And we might as well introduce Joe while he's here as well, sitting there. There we go. And the most important person for the registry is actually Katerina. So, Katerina, you need to actually stand up, I think, and make sure everyone can see you. There we go. Right. Okay. So Katerina will be having she's got a desk set up outside. So if what you hear now is of interest to you, or if you'd like to know more about it, then please come and see Katerina or myself, and we'll be outside later on, and we'll be here all day today. So the idea really is really to create a cohesive registry of individuals who have got alterations in the PTEN gene, who've got PHTS. And the idea behind this is that it's a rare disease, and for many years we only knew about a small number of individuals, and we get a very biased opinion about the condition because it's often those individuals who may be the more severely affected ones that seek medical attention. So it's very hard to get an accurate picture of exactly what this condition causes and actually what the risks are associated with this condition, or risk of complications. And particularly, my particular interest is hereditary cancer and how genes can cause cancer. So I've sort of come in it from that angle. But others in our group have other interests as well. So Catherine, for example, is very interested in the the neurological aspects of it and the developmental aspects of it in terms of the brain in children. So we look at this from different angles, and I think that's really important for you to realize that if you register for this, then we will be analyzing your data not just from one aspect, but from different aspects, and different experts will be looking at it in different ways. So that in a way maximizes the benefit from it. And the ultimate goal is to improve patient outcomes, treatment, and management of this condition. And it's a challenge because it's rare, and therefore, to actually get enough, our information really depends on good quality data and ideally large amounts of it. And that's really, the reason behind the registry is to collate all this information and put it all together. And from that, you can develop multidisciplinary approaches and you can help predict what complications may arise, and you can try and intervene before they arise, or pick them up at an earlier stage, essentially. There's also the opportunity on the back of this. Once you've got this set up as a foundation, you can then, for example, run virtual clinics. So if someone has a particular issue that we're not sure is related to PHTS, they could, contact we have this network of experts now in the UK that we could then feed into. So it may be that the local team hasn't come across this before, but then, asking through this network of experts, we can find out whether, someone else has come across this particular issue, and we can work together essentially to make sure that we get the best care for individuals who've got the condition. So a lot of the time, as I said before, my focus is very much on hereditary cancer, and a lot of the time, a lot of my time has been spent working with colleagues across Europe, essentially, to develop guidelines to make sure that we're offering the best surveillance for this. And this is all relates to adults, essentially, because the cancer risks appear later on in life, in adulthood. And the idea is really that if you can offer screening, surveillance for these cancers and pick them up earlier, they're much more treatable. And they may, for example, just need treating with surgery rather than things like radiation or radiotherapy or chemotherapy. So the whole idea here is about early detection, but there are also downsides to screening as well, because you can do too much screening, you can pick up too many things that may not be relevant, and you can then create additional problems where there weren't problems before. So there is a balance here, and we spend a lot of time trying to work out what the optimum balance is. And again, that involves discussing with lots of experts. This is at a European level here, because everyone again has different knowledge about how these cancers may arise. So I guess one question is, why is it so hard to actually estimate the cancer risks in this condition? And there are a number of reasons for this. And the first and most obvious one is that because it's rare, it's actually very difficult to get enough data together to make an accurate assessment. So many of you will have heard of the BRCA genes, BRCA1, BRCA2, Angelina Jolie and all that. I mean, I work in that field as well, and we are dealing with, our data there is based on about 20,000 carriers essentially, whereas in PTEN the biggest study is about 400. So you can see where the issues are, and even with 20,000, then we have some really good estimates, but there's still some questions that we have, even with that large data set. So by the time you get down to small numbers, it does become much harder. So that's by far and away the biggest problem. And the only way we can solve that is by systematically collecting as much data as we can in the UK, and then also pooling it with other countries as well, with Europe or US, to make larger data sets. And these are all anonymized at that point. So we're not sharing any personal information. We're just sharing data points, if you like. That's one big problem. The other problem is something called ascertainment. So, what happens is that someone, may get diagnosed with cancer. So, for example, if we've got a pointer here, yeah. So, for example, if you took, this young woman here, she may, for example, she could develop thyroid cancer at a young age. Someone did some genetic testing, they find a PTEN mutation, they find out that she's got PHTS. And that looks bad. I mean, if you just take that one person, it looks like there's a very high risk of thyroid cancer associated with this condition. But then, you test her brother, and you find out that he's also got the gene, and he's completely healthy. And then you go and test the parents, and you find out that the father's got it, and he's also got the gene alteration, and he's completely healthy. And suddenly, it doesn't look as bad as we thought originally. And so there are different ways that you can do the studies. The early studies only focused on the individuals, like would only focus on one person, and therefore the risk estimates were often very high. The more recent, better studies, what we do is we either ignore the first person, or we reduce using statistics. We make this less important when we're doing the calculations, and that means that we'd end up then with more what we think are more accurate estimates, because we're not so we're not being biased by the way that this family's come to light. And what you see is that the estimates go down, understandably, because that's and those are probably more realistic estimates. So we start off with high estimates of cancer, but actually over time, as we learn more, we get more data, and we sort of refine the statistical methods. The risk estimates are going down, and we see that in many conditions, in many rare conditions that we come across, and we're also seeing it in PHTS. So that, in a way, is good news. But we do know that there are definitely some increased cancer risks associated with these conditions. So, but that's when you go back in time and you look at some of the earlier papers, the numbers there are much scarier than they might be now, with the more recent data. And so this is what we're currently suggesting. We know that the main risks in females are breast cancer, and actually there is a very good surveillance program already set up for that for the NHS, because actually there are other hereditary conditions that cause breast cancer. So we've managed to sort of put PHTS, group that with all those other conditions, and the surveillance that we offer nationally is actually pretty good for that. And that doesn't probably need too much tweaking. I think it gets more complicated when we're talking about some of these other conditions, particularly thyroid cancer, is difficult because we are still somewhat unsure about what the right age is at when to start. And typically we've said 18 here. There's been a move internationally to reduce this down to 10. We are still sort of mulling this over, and I think we may end up being quite vague about this. And currently we're refining the European guidelines, and we're saying probably between 10 and 18, but on a but at a decision based on a local decision essentially. And the problem is with the thyroid that often, as well as cancer, that you get lots of benign lumps in the thyroid. So, and it's quite a difficult organ to look at properly to scan. So once someone starts scanning them and they see a few lumps there, they're not sure whether it's something, that is just benign or whether it's actually something that needs investigating. So you can end up then having surgery, possibly unnecessarily. And when you start doing that in young, in children, the consequences of that can be quite significant. So that's why there's quite a lot of hesitancy about that. And this remains controversial. I know Catherine, who I introduced previously, has got a very big interest in this in terms of how to optimize surveillance. And that's something that we're currently have been talking to the foundation about, whether or not we can do a study in the UK about how that might, can we come up with the right answer here for that question? I think that's really still one of the hardest ones. For kidneys, we have been saying they should have individuals have a ultrasound scan every 2 years. The current evidence is that maybe that may not be necessary. Maybe the risks of cancer may be lower than we had previously thought, just based on what I'd said in my previous talk. So we may be revising that down, and we may just say that you need a baseline scan of at sort of 35, 40, and then possibly no further scanning after that. But these are, we like to refine the guidelines according to new data that comes along, and sometimes that means reducing the amount of surveillance that we've offered previously, because we realize actually that amount of surveillance isn't necessary. And this could be a case in point there. For some of these other conditions like bowel cancer, melanoma, the risks also appear to be quite low. Already we were offering, not too much surveillance for those. And similarly for endometrial cancer, again, the risks are probably going down somewhat based on the newer new evidence that we have. So that's the theory behind it. But what happens in practice? And this is work that Joe's done. So he's talking to you after me, but he was kind enough to let me share this slide with you. And so what we did was, we're based in the East Anglia region, and we have a population of about 2.5, 3 million roughly. And we wanted to know, how many of our adults with PHTS are really getting the surveillance that we're recommending. And so we basically did an audit, did a survey of all our patients. We reviewed their clinical notes. And you can see that for some of the things like breast surveillance, we're doing very well, because a lot of that is already very well coordinated anyway, and there's a clear framework in place for that. And then, for some of these other things, we're doing okay. And maybe but we've also flagged some areas where it's a bit harder. So 9 out of 13 are having the thyroid. 10, the skin is a bit more difficult to know, because that's not something that we're organizing. We are referring them to skin experts, and sometimes they don't write back to us, or we don't know exactly what's happening. So this is really a bit more real world data, essentially. But the bottom line is, out of the 19 adults we have in our region, we're not, most of them are getting a reasonable surveillance. And again, the main focus really is breast in females, and for that we're doing reasonably well. And maybe we could be doing a bit better on the thyroid front. But this is like a benchmark, and we can see it helps us to know where the weak points are, where we need to improve. So these are the adults. We know that overall we've got 35 individuals in our region, essentially. So that's also a useful number to know. And that also reflects partly when we're talking about the registry itself. So this is the work that Katerina's doing. As of this month, we've got 100 and over 120 patients now on the registry, and you can see the female male breakdown there as well. And the 54 of whom are children, 75 adults, and you can see it's a good spread throughout the UK. But if we know that there must be many more, because if you've got 35 in our region of 2.5 million, that means there must be at least 700 across the UK. So clearly, we're still missing a sizable chunk of individuals. And I think Tom's going to present some more information about this, about how we think actually these gene alterations are much more common than we previously thought. But that gives you a snapshot of where we're up to with the registry at the moment. 129, it's not a bad number, but I think there's still a lot of room for improvement there. Um, and in terms of what it involves to take part, it's really a lot of this is just really about answering questions. We have questionnaires, which are really will ask you about different manifestations of the condition itself, and we'll ask you for your permission for us to access your local medical records. As I said before, we anonymize everything and we pool it all together. So when we do share data, it's completely unidentifiable to anyone. For example, if we're working with people from Europe, all we do is we give them a big description of what we've got in the UK, and that way it's a safe way of bringing together large sets of data that helps us then better answer the questions that we have. And these research studies are both have full ethical approval as well. Now, what we have done is shared information or worked together with collaborators, particularly Karaseng, who sadly passed away last year, and Tom will speak more about that. And also, people in the Netherlands have been very collaborative as well. So particularly Nicolie Hubrugge and Janet Vos, they're in the Netherlands, and through them, we've been able to work very well together with other European countries as well. Nicolene's actually retired now, but she still emails me a lot. So I don't think she's that retired. I don't think she's that retired. But anyway, there are other people there who she's been a fantastic collaborator and researcher, and she's built up her own team as well. So I'm sure that collaboration will continue quite nicely. I think a very exciting thing that's happening here in the UK, or certainly in England, I would say, is that we've got this new thing called the National Inherited Cancer Predisposition Registry. And this is being launched on Monday. And after a lot of hard work behind the scenes, and the idea is that actually this is now an NHS funded initiative. And the NHS does gather data on individuals, which is really designed to enhance the management and to know, the NHS needs to know what resources it needs and to put them where. And it's always collected data on people diagnosed with cancer. And more recently, it's been collecting data on individuals who have a genetic predisposition to cancer, and PTEN falls under that. So the point here is that the individuals don't need to consent to this because it's within the NHS essentially. And what's happened is we've developed a national registry which is focused on these cancer aspects, which covers many, many different genes, about over 100 genes. And all the clinical genetic services, so these are all the dots up and down the country, can now contribute to this registry. And it really serves two purposes. One is that it will allow us to better measure the surveillance that people are getting and also to request the right surveillance as well. So this is going to bring everything a bit more interconnected. And also it will allow us to identify more individuals throughout the UK. Now, because this is NHS, we can't, we still need to then seek out those individuals and consent them for research. But at least we'll have a better idea of how many people there are, certainly in England. So I think this is going to be a big step forward, essentially. And it was generously funded by the NHS. I know we hear a lot of bad things about the NHS, but actually I think they did come up with a good priority here. So this is done together with the UK Cancer Genetics Group and the NHS National Disease Registration Service. And these are the genetic centres that are taking part. We know that we're not quite sure what's happening in the devolved nations. And I guess for now we have, because of the governance issues and the way the NHS works, it has been very much focused in England. But I do hope that typically what happens in England that will then get copied or adjusted maybe a little bit for the devolved countries, and they will replicate what we're doing here using a similar infrastructure. So I do hope that eventually this will cover the whole of the UK. But I think this is a good start, and I'm hoping this will be a boost for our registry as well, which is much more. I mean, this is a good registry because it's looking at it doesn't serve the same purpose as what we're doing essentially. So I think what we can do is use this to enhance the recruitment to our registry. Again, if you have questions about this to me afterwards, then feel free. I'm here all day. We can talk about it. And then finally, our website. Sorry, it was actually down for a long time because we had a cyber attack. I think our cyber attack was much worse than the M&S one because we've been down for like it was a good 12 months. It was but I can really sympathize with the M&S people. Whenever I go past and see the empty shelf of sandwiches, I can understand why it's still empty after all this time because it is absolutely destroys everything and is really debilitating. And luckily, when we had our cyber attack, none of our confidential data was uh, taken. So so our security service systems worked to a point, but it did mean we've had to rebuild all our networks, all our websites. And it meant for like many months I was having to get up and walk to the printer with a USB stick to print stuff off, which did wonders for my step count. But it did remind me every time that actually, this is a how vulnerable you can be. Anyway, needless to say, now we've got the most tight security you could possibly imagine. So tight that even we can't access much of it. But the website is now back up and running. Probably needs a bit of a revamp, but at the moment, I think we're just pleased that it's there again. Actually, that was a major achievement. And these are our contact details, and I'd just like to thank everyone that's taken part so far. And again, Katrina and I and all the others here around all day, feel free to come up to us, ask us any questions. We're always really happy to talk to you. And it's great. These days are great for us as well because it just reminds us, why we're here and why our work is important as well. Because sometimes you can get a bit removed from it, and this brings us back down to the reality of it. And I always leave these meetings feeling, fully enthused again, and happy to carry on with the work that we're doing. So thank you very much. Yeah. Oh, yeah, Joe's up next. I'll introduce Joe. He's a clinical lecturer in our department, and he's doing some really interesting work, looking at more the basic science understanding of what the mutations do at the cellular level. And he's one of our bright young stars. So you'll see much more of him in the future, I'm sure.
Thank you, Mark, and thank you for having me here today. It's really exciting to be here. It's the first time I've been here at this event, and it's really a privilege to be here. And I'd like to talk to you today about an exciting new study that we've been setting up in Cambridge. We're in the early stages, but I'm excited to show you what we've done so far and what we now have available. So as a way to introduce myself, I'm an NHS doctor. I work in the same genetics service as Mark and Katerina. I also spend half my time doing research at the University of Cambridge and a research institute just outside Cambridge called the Sanger Institute, so just a few miles up the road. So I'm going to be talking about the cancer risk associated with PTEN. And this is a diagram some of you may have seen before. And what it summarizes is our current understanding of how cancer develops in any individual. This is not specific to PTEN. And the way in which it occurs is these stars show the different mutations. So these mutations that occur in our DNA. This happens the whole time. This is a natural process. It's a slightly scary thought that's happening to all of us as we sit here right now. It's a normal process. Thankfully, most of these get repaired. So most of these are repaired by our bodies, but some will slip through. So if you look at our normal tissue, what you'll see is that you'll have these mutations sitting there, and some of them are cancer associated. And if they build up to a certain level, that can then turn that cell into a cancer cell. And then when the cancer cell, can you see my cursor? I seem to have lost my cursor. Here it is. As the cancer then develops, it then acquire further mutations, and we get to the point where it's then detected. We give treatment, but unfortunately, a certain number of individuals with cancer will then go on to have spread of their cancer and relapse, and that's where the real difficulties begin. And a lot of research today has been looking at this end. So looking at treatments, trying to understand cancer once we've found it and how to best treat it. And there's been great progress in this area with new treatments. But my real interest is much earlier. It's here, the normal tissue and very early stages of cancer. Because really, if we want to prevent cancer, that's what we need to be focusing our efforts on, is either preventing it from forming in the first place or catching it at a very early stage when it's its most treatable. So looking here at the normal tissue and here at this very early stage. So to find ways that we prevent cancer formation and treat earlier, we need to better understand the early changes in normal cells before cancers develop. And this is my interest. This is what I want to try and figure out. So for me to do this, I need to give you a little brief background to how our normal cells work. So here I'm giving a very simple diagram. Those circles are 9 cells, and the square is any organ in your body. So those 9 cells will sit there and they will compete with each other. It's a battleground. And some of these cells will acquire mutations, and that red cell there has acquired a mutation. For those that are more visually minded, you can think of it as this patch of cactus in a desert, which ChatGPT did a lovely job of creating for me. And in one of the cactus plants had acquired a mutation and become a tree. But in this environment, in a hostile environment of the desert, the cactus is much better adapted. It's much more likely to be sustained within that environment, whilst the tree is at a disadvantage. So what happens in this situation is the tree is lost. What we can do is change the environment now. And in each of our tissues, there's a slightly different environment, and in each of us, there's a slightly different environment. So if you take the lung tissue of a smoker versus the lung tissue of a non-smoker, the environment changes. So that same mutation again, the cactus suddenly is very out of place and is not as adapted to the environment that it finds itself in, whilst the tree is very well adapted. So that same mutant cell can then expand. And what you then end up with is a patch of mutant cells, and we call this a clone, so a mutant clone. And we have thousands of these in our bodies, I'm sorry to say. And most of them will cause us absolutely no problems. But some of them, if they grow big enough, can then acquire additional mutations. So they not only have the first mutation that the red cell had, but they then have extra cancer-associated mutations. And this is when it starts to get risky. So this is represented here by the black cell or a very big tree. And these very big trees, the black cells, have a really strong advantage in their environment and they can expand quite rapidly. Now, the example I've shown here is quite simplistic, as I'm sure you can see, and the actual picture is very complicated. So we have a lot more than 9 cells. We have billions upon trillions of cells in our bodies, and they're constantly competing with each other and changing. Some are being lost, some are being gained, and the environment itself is also changing. So if that smoker then quits smoking, the environment changes again, and certain cells that would have had an advantage in the smoker's lung suddenly is at a big disadvantage, and you lose those cells. And up until about 10 years ago, our ways of looking at this patchwork of these different mutant cells, these clones in our normal tissues, was nigh impossible. But over the last, and it was a situation like this, we couldn't see the different trees for the forest. The way in which we're looking at our cells was just too big a picture. We're too far out to be able to say with any certainty which mutant cells, which mutant clones were present in a person's normal tissue. But over the last 10 years, we've made massive progress in this area, and we have lots of new technologies that allow us to then separate these single cells, these different mutant clones, and look at them in high detail. And the 2 main technologies which I'd like to draw your attention to, and which we're proposing to use in this study, is laser capture microbiopsies. This is where we take tissue and we slice it into very thin slices using a very sharp knife, essentially called a microtome. And when we put it under a microscope, and then we take a laser, it's honestly like something from a James Bond film. It's amazing when you're using this. You can see each of the cells individually and then use a laser to cut out these microbiopsies. And what we're able to then have is a small cluster of 100 to 1,000 cells. And that allows us to do really high detail analysis of those cells. The second technology is something called NanoSeq, which I won't go into the detail here. I'll be outside if you want me to bore you about NanoSeq, I'd happily do that. But essentially, it's a very high resolution DNA sequencing technique that again allows us to see these cells in each of their individual forms and pick up these cancer-associated mutations in normal tissues. So how do we take this into the clinic? How do we use this for patient benefit? It's very useful so far being able to understand the biology of different tissues, but how can we actually apply it? So what I'm proposing, and what others are also thinking about at the moment, is taking individuals and looking at their clones, their clonal landscape, the different mutant cells that they have in their bodies, and trying to identify those that are highest risk of developing cancer going forward. So here you've got these 6 individuals, and we've used our new technologies to try and identify the different mutant cells within their bodies. And here you can see these 2 individuals in the center. Slides malfunction there. The red box should go around these 2 individuals. They have more black cells than others. They have more of these mutant clones than what would be expected. And that's really helpful because it means we can pluck out those high-risk individuals and either put them on more intensive screening programs and surveillance, like what Marcus talked about, or try and develop new technologies to try and detect these cells using technology other than what we're using, such as NanoSeq. Equally, for these individuals, they may provide some reassurance. We could say, actually, we'll continue on the same screening program you're on currently, or even potentially scale it back slightly. But thinking even further ahead here, for a few years down the line, wouldn't it be amazing if we could take these high-risk individuals and give a treatment that directly targets these high-risk cells and removes them from the body entirely? We're a long way from there, but using these technologies and applying them in the right way, it's something we could certainly start working towards. And this is where people like you, families like you come in. What we need to start doing is to collect samples, to biobank samples from individuals which we know have an increased risk of cancer during their lifetime. So we've developed a study, it's called RECCE PTEN, and it's co-led by myself and Rahela Rabari, who's a scientist at the Sanger Institute, in collaboration with Katerina and Mark, who you've heard from already this morning, using the National Registry to identify individuals that would be appropriate for our study. And we've set up a clinic in Cambridge with a dedicated research nursing team and dedicated research clinic space, where individuals who are interested in being involved with the study can come and meet with us in Cambridge, and we can take samples, donations at those clinic appointments. At present, to be eligible, you need to have a germline or mosaic variant in PTEN. We are also able to recruit unaffected family members to the study as well. We need to look at other individuals who don't have the variant, so family members who are unaffected would also welcome to the study. And the key thing is that the individual needs to be able to consent to the study, or a parent should be able to consent on their behalf if they're under the age of 16. At the moment, we're only open in Cambridge, though we are hoping to expand to other sites in future. The way in which the study works at present is that we would ask individuals to come to our clinic, and if they're happy to go ahead, consent and sign the paperwork at that clinic appointment. And then we'd ask for the donation at present for a blood sample, a cheek swab, a urine sample, and also an optional skin biopsy. It's a 5mm piece of skin. We use a small amount of local anesthetic to take that sample. And then we'd invite you back in a year's time to donate the same samples again. In the meantime, we also asked if anyone was undergoing any sort of procedure, whether it be related to cancer or not, to also inform the study team, and we try and biobank surplus tissue from those procedures as well. So in the very early stages, we are recruiting. We've got a good number of individuals on the study already, but we're always looking for more, and we'll hopefully be going to be able to expand going forward. So if that's something you feel like you want to be involved with, be more than welcome to get in contact with me, and my email is there. Equally, if you have any feedback, comments, anything that you'd like to discuss about the study, or feel like things we should be doing differently, or things you'd like to see included in the study, do get in touch. I'm more than happy to answer any questions that you may have. So thank you. I can, yes. So I've met Tom briefly online before, and he leads the PTEN Research Foundation, and he'll be giving an update now. Thank you, Tom.
Well, great. Thank you very much for the introduction, and thank you very much to Kelly and everybody at PTEN UK and I for the introduction. We always very much enjoy coming to these meetings and interacting with families. What I wanted to do today is give you a little bit of an update in terms of the work of PTEN Research. And here is a quick overview of what I'm going to be talking about today. But as Mark alluded to, before we talk about the foundation in a little bit more detail, I just wanted to pause for a moment to honour the memory of Professor Karasang, who very sadly and prematurely passed away last summer. I think everybody in this room and the whole of the PHS community has a lot to thank Karis for. She was a real pioneer in the field. Back in the 90s, she was a member of the key team that identified variants in PTEN gene were associated with Cowden syndrome. And she also was able to demonstrate the role of the PTEN gene, normal PTEN gene, in terms of protecting against cancers. And over the preceding decades, with her team first at Ohio State University and then at Cleveland Clinic, and also collaborators across the globe, she really led transforming our understanding of the biology of PHTS and the diverse manifestations that people with PHTS experience. Speaking very personally from PTEN Research's perspective, Karis really had a seminal role in helping to drive our strategy, and she was always incredibly generous with her time. In fact, actually, people joked that, if you sent an email to Karis day or night, during the week or the weekend, she would always respond back within a few minutes. And actually, that was my experience as well. And we were really privileged to continue to work with her up until her death last summer. So again, very sad news, but we want to continue with the work that Karis did and really protect and build on her legacy. So switching gears a little bit now, I wanted to provide a quick overview about our organisation and then some of the things that we have been doing in the last couple of years since the last patient meeting in Oxford. So we're a UK-based charity. We were formed in 2017, and our exclusive focus is to fund medical research into PHTS. And our mission is to fund and facilitate that research that will lead to new and better treatments for individuals with PHTS in the future. And our vision is to have those therapies available to patients within the next 10 years. We're a really small organisation. We're just 7 people, semi-virtual. We don't have any offices. And I'm joined here by my colleague, Carola. Carola, do you want to just... So we're here for the whole day, and please do at any point reach out to us to talk a little bit more about what we're doing and how we're interacting with the PHTS community. We'd love to hear from you. This is a little bit of a busy slide, but I just wanted to give you a sense of the type of work that we're doing and the scope and the breadth of our activities. So first of all, of course, we know PHTS is a rare disease, and because it's a rare disease, we want to work with the most innovative and experienced researchers, regardless of where they're geographically based. So although we're a UK-based charity, we currently are working with hospitals and universities, not only in the UK, but also across Europe, North America and Australia. And we've also had collaborations in the past with organizations in Japan. We don't have our own laboratories, so we are very much dependent on partnerships with academic researchers in hospitals and universities, and we also do work with contract labs, and I'll talk a little bit more about that in subsequent slides. One thing that we're also pretty interested in is trying to raise the awareness of the unmet medical need that people with PHTS experience to researchers in the pharmaceutical industry and biotechnology companies. And that's for a number of reasons. One is that those people are really experienced in developing drugs in rare diseases and in cancer. And also in the future, we really want to be able to not only leverage their knowledge and their expertise, but also their funding to help actually develop new treatments for people with PHTS. And we think those kind of collaborations are really important. A key metric, I think, of our work is actually publishing the data that has been generated as a result of the studies that we fund. And actually, a prerequisite of all of our grants is that the work is published. So that means that researchers around the globe can benefit from the knowledge that has been gained, and it's there in the public domain in perpetuity. And so far, our funding has been acknowledged in 54 different papers, and that number is constantly growing. As I said to you before, we're a very small organisation, just 7 people, and we can't hope to know everything there is to know about this very complicated condition. And so we rely very heavily in terms of interaction with researchers and clinicians across the globe, including the folks that are in this room today, to drive and refine our strategy as new data becomes available. One piece of work that I'm really passionate about personally is actually developing tools to measure the impact of a new treatment for PHTS on people who are affected. And the reason why that's important is if you can't measure what success looks like, it's very difficult to set up a clinical trial to test a new medication. And so this is what we call a clinical trial endpoint. And actually, that's a surprisingly complicated thing to do. And it's something that we've really invested quite heavily in. And I'll talk to you a little bit about a case study later on in my presentation. And the final thing that actually was a collaboration with Mark and also with Professor Eng is that the foundation partnered together to obtain what we call an international classification or ICD-10 code for PHTS. And it sounds kind of very dry and very administrative, but this is a code that is unique for PHTS that can be put into an individual's medical records. And that's really helpful because actually, once it's in somebody's medical records, you know that they have a definitive PHTS diagnosis. It helps you estimate the prevalence of PHTS, the frequency of PHTS. And when clinical trials come, it helps identify those individuals. And actually having that code actually really helps future-proof and support research in the future. And just to sort of say that there are probably about 6,000 to 10,000 rare diseases, and maybe 200, 250 have their own ICD-10 codes at the moment. So actually by having those ICD-10 codes for PHTS, we're ahead of the curve to allow us to support future research in moving forward. So, a great thank you to Mark for his help on this. I think this slide probably doesn't need a lot of background and context. Clearly developing new treatments for any condition, particularly a rare condition, is a collaborative effort. And clearly we want to work with a whole group of individuals who are involved in therapy development. And clearly input from yourselves as patients and family members of people with PHTS is absolutely critical to that, as is the input from academic researchers and expert physicians. We want to get input from pharma and biotech, as I said before. And then also another important gatekeeper are, of course, people in health authorities. So the MHRA in this country, the Food and Drug Administration in the US, because it's those organizations that ultimately are going to be the groups that will make the decision as to whether a new treatment for PHTS is licensed and is available for doctors to prescribe. And so we have increasingly had a number of interactions, particularly with the US FDA and more to come. Mark has already talked about the registry, and we're very proud to say that the foundation is continuing to support funding of the registry until at least 2028. And so obviously, if you are interested in participating in the registry and you haven't done so before, please talk to Mark or Katerina during the course of the day and to see whether participation might be right for you. I wanted to switch gears now and talk a little bit about how rare is PHTS and why is that important? So historically, the frequency or prevalence of PHTS has already been cited as being about 1 in 200,000 or 1 in 250,000. And so that would equate maybe to about 350 people in the UK having PHTS. But I think it's important to stress that that is really just a very rough estimate. It's based on a trial that was undertaken in the Netherlands in the 90s. And it actually was focusing on people with Cowden syndrome rather than people with PHTS. And we know that actually diagnosing people with PHTS is really quite hard because people have such a broad range of different symptoms. And many of those symptoms overlap with other common conditions. Some people might not be diagnosed until later in life. Some people might go their whole lives without ever having a PHTS diagnosis. And so these estimates of the prevalence of PHTS are really hard things to do. And so actually, in April of this year, there was some new data published which focused on two new data sets. And this was a combined data set of about 700,000 people. And this estimated a new prevalence of PHTS of around 1 in 9,000 to 1 in 13,000. And so, that's about 20 times higher than was previously estimated. Now, where did that data come from? So there were two research studies, one in the UK and one in the US. And actually, people volunteered to participate in those studies. They didn't have to have any particular condition diagnosed. They didn't have to have any symptoms. And so actually, they just volunteered and were given a sample that was which was genetically tested. And what we hope is actually to a greater or lesser extent that the people who volunteered to go into that study represent the background patient population, the population of the US and the population of the UK. Probably some biases, but actually not too bad as a representative sample. And I think the fact that actually both cohorts, the UK cohort and the US cohort, came up with such comparable numbers gives us some really good sense that actually this may be much closer to the true prevalence of PHDS. I think one thing that is really important to underline is that PHTS clearly still remains a rare disease. And the other thing is that we don't know within those groups actually how many of the people actually had a PHTS diagnosis. It may be that a lot of those people don't have a PHTS diagnosis, and it might be actually that quite a lot of those people actually don't have any symptoms associated with PHTS. So although it's interesting and very valuable, it asks new and important questions that we hope to address by doing further studies in the coming years. But this is a really important and valuable information because it helps healthcare systems really plan and budget appropriately for PHTS, because it indicates that there are probably a lot more people with PHTS than we previously thought. Hopefully, it will also stimulate interest from research groups, both in hospitals and universities and the pharmaceutical industry, into PHTS. And in combination, I hope that will drive better outcomes and treatments for people in the future. So again, I hope reason for a little bit of optimism. I wanted to spend a few moments now talking about a couple of projects that we've been working on within the foundation and providing grant funding support for, and where they are at the moment, and some key updates there. So this is actually a little bit of a study that Mark had alluded to in his presentation to try and better understand the risks associated with cancer, the prognosis and the impact of lifestyle on PHTS-associated cancers. The foundation had been working for many years with a group based in the Netherlands at Radboud Medical University under the leadership of Professor Nicolien Hugenbrugge, who you can see in the right-hand corner of the slide. And what Nicolien's team did is that they undertook a big retrospective study, and they collected data from multiple centres across Europe and actually collected data from over 500 people with PHTS, which is no mean feat because obviously we know PHTS is a rare disease. And as Mark alluded to in his presentation, that included data from individuals who had participated in the UK registry. So again, thank you to everybody in this room who participated in the UK registry. You supported this research with your data that was anonymized before it was sent to the Netherlands. And it has resulted in 8 separate scientific papers in the literature. And there's an awful lot of detail and data there, but I just wanted to share some of the key findings and key takeaway messages. So first of all, it helps us better estimate the cancer risk associated with PHTS. And again, as Mark alluded to, there are reasons why historical estimates might have actually been thinking that risk is somewhat higher than it may actually be because of ascertainment biases. And actually knowing the true risk allows that cancer surveillance for PHTS can really be optimized and made appropriate for those individuals in that population. So again, I know that work is ongoing to optimize PHTS cancer surveillance, both in Europe and in the UK. It also showed, and I think this is really good news, is that the outcome for people with PHTS who experience a cancer is comparable to people in the background patient population, people who don't have PHTS who have a cancer. So it appears to be very comparable. And that wasn't necessarily expected to be the case, but again, I think is actually relatively positive news. It did, though, show that actually, if you have had a cancer as a PHTS patient, it's really important for you to continue with your cancer surveillance, even if you've had risk-reducing surgery, because people with PHTS might continue to have additional cancers, so-called second primary cancers. So it's really important to continue with your cancer surveillance, even following a cancer diagnosis. And the final piece, which again, I think is something that's really actionable, that actually people can address themselves, is that the data showed that healthy lifestyle, increased physical activity, reduced alcohol consumption, reduced tobacco use is actually associated with a lower cancer risk in PHTS. And again, that mirrors what happens in the background patient population. But the fact that we know that that is very similar or broadly comparable in PHTS patients, again, I think is reassuring. And also it gives something that everybody can actually do and implement in terms of looking after themselves and implementing those changes in their lifestyle. The second study that I wanted to spend a little bit of time about is to develop tools to assess the thinking skills and behaviors in people with PHDS. And the reason why this is important is that, again, we would really like to support future research and clinical trials for new agents for people with PHDS and issues around autism spectrum disorder and learning difficulties. And at present, there's a real challenge because measuring thinking skills and behaviors in PHDS patients is actually really difficult. To do that at the moment, you broadly have two different types of instrument. So you can have surveys and questionnaires, which are completed by a caregiver, by a parent, by a partner on the individual with PHDS. But those can be very time consuming and can lead to people filling in those questionnaires, actually rushing through it or becoming tired. And there is this well-documented feature called rater fatigue, which actually means that actually, if you're asked to fill in a really long questionnaire or lots and lots of really long questionnaires, your ability to do that actually diminishes over time. The other thing you can do is actually ask the individual with PHDS to do a particular task, but that requires them to go to a specialist clinic and have a doctor who is specially trained or a psychologist specially trained to assess their performance in that task. And again, just to show you how complicated all of this is, in some of the early studies with PHDS, a comprehensive assessment of thinking skills and behaviors took about 6 hours per time point and required in-clinic assessment. And you can understand that's really only possible for the most motivated families and the most motivated hospitals. It doesn't really facilitate easy research. So what Professor Tom Frazier from John Carroll University in Ohio did was to set out to build some new PHDS specific survey scales and performance measures with the input of families, of people who are impacted, and PHDS experts, because we really wanted to understand what was important from families and from treating physicians in terms of PHDS patients. And this work has already led to a number of papers being published in scientific journals. And now I just wanted to have a big shout out now and really sort of big up the UK and thank, I know a lot of people in this room participated in this study. Actually, 64 people in the UK participated in the study, and many thanks to all of you for taking your time to do that, and also particularly to Catherine to coordinate that work out of Southampton. A huge amount of effort and work. And actually, that was significantly higher than the recruitment that was achieved by Cleveland Clinic as a world-renowned centre. So I think a real big up for the UK in doing that. What were the findings from this? Well, first of all, it's really quick. To do what takes 6 hours using conventional measures takes about 30 minutes using these new tools. It's all done at home, it's all done online, there's no travel, and there's no need to train specialist physicians or psychologists to do this. And then also from a more technical perspective, we were able to demonstrate that the data tools were accurate, they were consistent, and they measured what they were supposed to measure, what we call psychometrically validated. So this is really exciting. This enables future research to take place. So what's next? We're trying to now move the tools into a second stage of validation. So we're doing head-to-head comparisons to see how these new tools compare with the existing tools. So there's an ongoing autism spectrum disorder of people with PHDS that's being run out of the US, out of Cleveland Clinic. And we've built those new tools and the old tools into that, and we'll be able to see actually head to head how the two different types of tools perform. And we're also in the process of trying to set up a new trial of everolimus in the US. And again, that will use a combination of new and old tools to allow that cross-tool validation to take place. One piece of work that I'm personally really proud of is that we have made an application to the US Food and Drug Administration to a scheme specifically designed to validate these type of tools in rare diseases. It's a really competitive scheme. There were 13 places available over the period of 3 years, and we were really lucky to have been awarded a place on that scheme on our second application. So again, I think a piece of work that we're really proud of, and then we hope that will mean in the future that the FDA will recognize these tools as the standard for future PHTS research. And wouldn't that be great if we can reduce something that took 6 hours for PHTS families to 30 minutes, and nobody has to leave their home to do that assessment. And that, I think, really makes future research much easier and democratizes activities and making it much more patient-centric. Wanted to talk a little bit about PHTS diagnosis now, and I think it was Pete who said, how challenging it was, when you receive a diagnosis of PHTS, not only for the person involved, but also for their family. But there are also some benefits of receiving that diagnosis. It means that the person who gets a diagnosis can start to receive supportive care, and they can also start to have cancer screening, which we know is of benefit to those families. Also, other family members who might also be impacted could then consider whether having a genetic test is right for them. So actually, there are benefits of getting a PHTS diagnosis for the individual. How can we improve rates of diagnosis in a timely fashion? And we're supporting a number of projects in this area. And one of them, I think, first of all, goes back to the point that I made in the beginning of my presentation, that PHTS remains a rare disease. It's estimated there are 6,000 to 10,000 rare diseases. And so it's impossible for most doctors to really understand about all rare diseases. You can't train people to recognize all rare diseases. I think that thought process is just something that isn't viable and doesn't make sense. Can we use computers to actually do that in a much more volume way? Is it possible to train a computerized algorithm to look at people's medical records and look for signals or traits in the medical records that might identify that an individual potentially has some traits of PHTS, but is currently undiagnosed? And if that was successful in the future, you can envisage a situation where you run the algorithm on people's medical records, and a signal is sent to the GP, and the GP is asked to consider whether it might be right to consider having a discussion with that patient about whether further clinical assessment is required. Now, again, that all sounds very straightforward. I think it's really important that we don't want to obviously identify a very large number of people who subsequently don't have a PHTS diagnosis. We don't want to create that level of anxiety for people. And of course, genetic testing is very time consuming and very expensive. And so it does rely on building a computerized algorithm which actually has a high level of sensitivity and accuracy. The work that we're doing really follows on from some activities that were previously done by Harvard Medical School and Boston Children's Hospital, which actually identified that computer algorithms were pretty good at identifying children who would benefit from PHTS diagnosis. But also, actually, in some senses, that might be an easier thing to do because we know when people are diagnosed in childhood with PHTS, that's usually because they have a larger head circumference or they might have learning difficulties. And the people who aren't diagnosed with PHTS might have more subtle symptoms. And so it actually might be harder to identify those adult people. So we'll see in the next couple of years. But we hope that this approach could actually be utilized not only for PHTS, but for other rare diseases and reduce what we call the diagnostic odyssey. Just my final couple of slides now is to talk about what's coming next for PTEN research. So we have a couple of clinical trials that we're planning. These are currently both within the US, with US collaborative groups, with hospitals and universities. One is focusing on a trial for people with PHTS and social difficulties, and builds on the previous trial of everolimus that was conducted a couple of years ago. And the other one is in an unnamed drug focusing on people with vascular anomalies. And this really builds on previous work that we've done to try and repurpose existing drugs that have already been approved in other patient populations and create evidence to support having a clinical trial. I hope to be able to make some public announcements soon as we have all of the regulatory and funding approvals in place. And particularly, I stress that unfortunately we are under confidentiality agreements with some of our collaborators, and I'm unable to share more information today. We're also looking to try and think about how we can further repurpose existing drugs for PHTS. One piece of work which is ongoing and we hope to reach out in the next couple of weeks is actually using AI approaches to mine the scientific literature, pattern databases, genetic databases to see if there are existing drugs that actually, from an electronic perspective, there might be some evidence that could be of value for use as a new treatment for PHTS. And in partnership with Boston Children's Hospital and a commercial lab called Charles River, we're also planning to screen the entire library of drugs that have been approved within the United States in PHTS-specific models. So in the past, we focused on a very small subset of particular molecules where we thought there was some biological relevance. And now we're going much broader. We're going to screen absolutely every drug on nearly 4,000 different molecules to see whether there is a possibility that some of those could be repurposed for PHTS. Now, clearly, that's a very long way from a clinical trial, but it's trying to turn over every stone to try and identify new treatments in the future. So with that, I will close and just say a big thank you to everybody in this room for your help and support. We couldn't do it without you in terms of your support of the registry, in terms of your support for the development of the neurobehavioral tools that I talked about in my previous slide. And if you'd like to hear any more about the foundation, please do come and talk to myself or Corolla at any point during the day. I think now I'll be handing back to Kelly because I think it's nearly lunchtime.